Hi, AP Bio. This is me going over day three of our review work. So first I'm going to start with some AP exam tips, then I'm going to do a mini lecture on Unit 4, and then I'm going to describe the homework for today. First, some AP exam tips. I did, if you remember, a Zoom FAQ session. Shout out to Sam and Rosie for showing up. The link to the recording of that is here. This link here is my working calendar that I'm using to plan. So if it helps you to see an overview of what's coming up, you can click on that. And I strongly recommend, if you haven't already, that you click on this link because there you will see little shout outs from each of you to your classmates. And it is one of the most adorable things, definitely one of my highlights last week, for real. Yeah. Some AP review tips. So don't forget that there are daily lessons every day at 1 p.m. From College Board. Just a spotlight on 2 upcoming lessons. This one I've linked right here is going to be on photosynthesis. This one right here is going to be on cellular respiration. I know those are 2 topics that you guys often request extra review time on, so I would strongly recommend checking those out. And then also remember that on the weekends, the AP Bio teacher community has review sessions that they are running. And if you click here, this will give you the link to all of them, but this one in particular, I'm spotlighting today because it is on the content from our Unit 2. So again, that will include photosynthesis and cellular respiration. So lots of opportunities to get extra help with those topics. This is just here as a reminder of what units, like what topics I'm talking about when I'm referring to unit whatever. So today, the mini lecture is going to focus on Unit 4, which is Mendelian genetics and heredity. Remember, that's College Board Unit 5, just in case you're looking at the external resources. So get a pen and paper or something to take notes with, pause the video, and then come back so that you're ready to take notes. So Unit 4 was all about heredity. And we started out super brief, just doing a review of basic Punnett squares. So that's what we see here. This is basic Punnett squares. We see a cross between 2 heterozygous individuals gives us the classic 3-to-1 ratio. And that's because the dominant trait overshadows the recessive trait. We only see the recessive trait when we see 2 recessive alleles together. Okay, so that's basics that you learned back in 10th grade as well. But then we started doing things like dihybrid crosses. So here we have a dihybrid cross. A dihybrid cross would be when you take 2 individuals that are heterozygous and cross them together, but they are heterozygous for 2 traits instead of here, where they're heterozygous for just the 1, which would be flower color. Here, this P is heterozygous for the round versus wrinkled shape and for the color of the P, yellow versus green. So this P is round and yellow. If we look here, so is this P, round and yellow, but it is homozygous dominant for both traits, which is why all of the offspring turn out round and yellow, because those dominant alleles overshadow all of the recessive alleles from this individual. So that's a pretty simple cross right here in this P generation. But then when we have our heterozygous ones, that's the F1, and we cross 2 of the F1 together, we end up with this 9-to-3 to 3-to-1 ratio right here. That's the classic dihybrid cross ratio for the phenotypes because of this Punnett square right here. So I know that dihybrid crosses have been challenging for some of you and then pretty easy for others of you. So this is a good place to really be honest with yourself and check, like, is this something that I really need to work on or not? And if it is, then put in some extra time, make sure that you're really studying it. If it's not something you really need to work on, there's probably other topics you need to work on more. So this is where you start to make those decisions. Yes. Okay, I'm going to continue forward, but I am going to talk a little bit more about dihybrid crosses in a moment or two. So next we talked about sex linkage. Remember, biologically speaking, a male in humans usually is XY and females are XX. And again, this is both usually and biologically speaking, not talking about gender. So that means that any trait that's on the X chromosome, males only have one copy of, which is why we see, for example, here with hemophilia, which is a sex-linked trait. If the mother is a carrier, she doesn't have it, right, because it's recessive, but it means that she can pass it down to one of her sons, because her sons will only have one copy of this gene, and so they don't have the opportunity to have, as we see here with this daughter can have a dominant allele overpower her recessive allele, whereas the son here cannot. Remember, these are 4 possibilities, but it means that there is a higher likelihood that a male will have a sex-linked trait than a female because of this phenomenon that we see here. Then we applied these concepts to our virtual fly lab, and we did some chi-square with that as well. This is a problem that you might recognize from some of your quizzes and exams. Here we see the F1 data and the F2 data, and what you needed to do was figure out first what the parents' genotypes were, and second, do a chi-square for that. So I would recommend you pause the video right now, try to work through this problem, and then I'm going to talk about it. Okay, hopefully you paused the video and tried to work it out. So I'm just going to talk it through now. So the parents would have, first of all, this has to be sex-linked. And we know that because we see that the male is, for the wild type, has 0, whereas the female has 45, and then same, we see that with the white-eyed. So when we're seeing these zeros and this huge difference between sexes, that tells us it's sex-linked. So we know it's sex-linked. Then next we have to figure out what's dominant, what's recessive, what does the dad have, what does the mom have. So the dad is wild type, and that tells us that so then if we do a potential cross, we can figure out that what the different genotypes of them are. Okay, so I had to just pause to pull up the key, but here's the key for this problem. So the parents, the dad has to have the dominant allele, and the mom has to be homozygous recessive. And the reason for this is, if we see this, if we look at this and we put it into a Punnett square, what we would see is that all of the females are going to be getting the big E from their dad, whereas all of the males are going to be getting the Y from their dad. This means that all of the females are going to be wild type, whereas all of the males are going to be getting the recessive trait from their mom, because they're going to have the recessive trait and the Y. So again, all of the females are going to be wild type because they're getting that from their dad, whereas all of the males are going to be white-eyed because they are getting that trait from their mom along with the Y from their dad. So that answers part A. Then for part B, you're going to do a chi-square analysis. And the first thing you have to do here is you have to do the Punnett square for crossing the F1. So taking this individual and this individual, crossing them together and seeing what we would get in the F2. So this is taking the heterozygous female crossed with the recessive male. Okay, so these are the kids of these two parents, and you're going to see that you're going to get a 1-to-1 to 1-to-1 ratio. What this means is you're going to have one female that's wild type to one female that's white-eyed to one male that's wild type to one male that's white-eyed. And so then you're going to do a chi-square where your expected values are all equal because it's all 1/4. So your expected values are 25, 25, 25, 25. And then you're going to work through and you're going to see that your chi-square value should be around 2. Then you compare that to the 3 degrees of freedom critical value, which is 7.82. And yes, we accept the null hypothesis. This means that yes, we did our Punnett square correctly, that it is in fact a 1-to-1-to-1-to-1 ratio. Okay, so then we went on to talking about meiosis. So this image I think could be helpful in showing you and just reminding you some of the key differences between mitosis and meiosis. The most important thing is that at the end of meiosis, we end up with 4 daughter cells that all are haploid, meaning they have half as much DNA as the parent cell, and that are genetically different from each other. So what I mean by that is that these 4 daughter cells at the end have gone through independent assortment and crossing over to make the gametes be each containing slightly different genetic information, even when we're talking about from one individual. So for example, my eggs are all genetically different from each other, even though they're all coming from the same genetic information, my genetic information. And what this means is that if we then think about sexual reproduction, so taking one individual's eggs, combining them with one individual's sperm, we end up with this functionally infinite amount of variety in the outcomes. And this is the reason that siblings can look so different from each other. So this is, again, independent assortment, which I'd recommend you review what that is if you don't remember. Independent assortment makes there be 2 to the 23rd possible combinations. Then we can multiply that by a second individual, right? 2 to the 23rd for the egg, 2 to the 23rd for the sperm. Those multiplied together, we end up with a huge number. Then if you include crossing over, which basically can randomly swap sections of DNA throughout the process of meiosis, we end up with just an enormous amount of variety that's possible. So even the same 2 parents, we can end up with vastly different offspring. And if we combine that with the concepts that we've learned in with the dihybrid cross, etc., that's where we got this problem. So again, I recommend you pause the video, try this problem, which you've seen a few times, and then I'm going to go over it. While you work on it, I'm going to pause my video too to find the key. Okay. So the first thing you need to figure out in this question is you need to figure out what would end up in each of these 4 gametes from this original parent cell, and this cell is representing the F1. So the first thing you need to understand is we're starting out with these 2 parents, right? And one parent is homozygous dominant, the other is homozygous recessive. If you cross those 2 together, you're going to end up with an individual that is heterozygous for both traits. This is a similar concept to what we saw here, right? We have 2 homozygous parents, we end up with a F1 that's all heterozygous for both traits. Now you're crossing those together. This is sort of like a dihybrid cross, but before it's asking you for the outcome of the dihybrid cross, it's just asking you to show what would be in the gametes of one of these individuals. So it's a heterozygous individual, which means they have all of the possible alleles, which means they're going to split up in all possible combinations. So this is what your 4 circles should look like. Each has 2 chromosomes because, remember, it has half as much DNA as the original parent cell. And you have all the possible combinations, big G with big D, little g with big D, etc. All possible combinations. And you should notice that these possible combinations are the same pairings as we see along the side of a dihybrid cross, right? 2 capital letters, a capital letter and a lowercase, the other option of lowercase and capital, and then 2 lowercase. So you see like a pattern showing up. And that pattern is created actually by independent assortment. Independent assortment is the reason why you get all 4 combinations and you don't just get big G with big D and little g with little d. You get all possibilities. Okay, so now we have this, and then it's asking for step 2, what would we expect in a cross between the F1, so this heterozygous individual that we were talking about here, and this individual. So instead of doing a dihybrid cross like this, it's saying you have the heterozygous individual on one side, but on the other side, you have a homozygous recessive individual like this. So what would you expect? So if you do that dihybrid, or that 2-trait cross, which is going to look different than this one, you'll actually end up with 1 to 1 to 1 to 1. That's because you're going to actually end up with each row is going to look the same. Okay, and that's because the homozygous recessive individual isn't going to have 4 different versions. It's just going to have one version the whole time, the little g, little d the whole time. So you're going to end up with 25% green dwarf to 25% green tall to 25% purple dwarf to 25% purple tall. Then for the last, it says that you see a proportion that's different than this, and in fact you're seeing, you're seeing that they are genetically linked. What would be the giveaway? And the giveaway would be if you see more individuals that are like the parents than you would expect. Because remember, if they're genetically linked, you're actually going to see a higher percentage of what the parental phenotypes were, because genetically linked means crossing over isn't happening. So you're not seeing that variation being created like we would normally expect. So there would be a greater percent having the parental phenotypes, or greater than 25% would be green dwarf and purple tall, because those are the parental phenotypes. Okay, so I know that was a bunch of information. Again, I recommend you watch the video multiple times, or also know that we're going to keep reviewing. You'll have some free response review of Unit 4 later this week. But for now, what are you going to do? You're going to do a case study on COVID-19. This is part 2 of the case study that we started last week. Just a little joke for you, referencing Unit 2 that we reviewed last week. So the Google Doc that you're completing and turning in today, so this is due on Wednesday, April 15th at 11:59 p.m., is this one right here. It's a continuation of your lesson from last week. The only thing I want to note is you have to answer all of the questions in purple. When you're using Desmos, you should be plotting the X should be days since first case. Your Y should be total number of cases. You're going to need to do that twice. So make sure you keep the graph up. What I did is I had 2 separate tabs of Desmos so I could compare them. You could also take a screenshot of one and then reuse that tab, but just don't just get rid of it, because you are going to need to compare the 2 graphs that you make. You're going to be doing another graph here for this data. And then the last thing I want to say is, if at the end of this, I know it's sort of overwhelming to do a lesson about what we're currently experiencing, but I also hope that it's helpful to get some actual data and some real information rather than just what we see in social media. But if at the end of the lesson you're left with questions about coronavirus or COVID-19 and you don't know where to find the answers, please email me your questions, because I would love to do my best to find the answer, or at least to find out what people are currently researching about that topic, so that I can include it in a future lesson, because I would love to be responsive to what you guys want to know. And I also I would be happy to help you try to parse out what's real versus what's fake in the news and media right now. And I want to remind you to always be skeptical of what you see on the internet, especially when we're talking about current events. That's really important. Make sure you're always looking at a trusted source. All questions are welcome. And I miss you guys. Bye.